Good morning, everyone, and thank you for coming to our fourth webinar about Discover Cell Shapes. My name is Laura, and I'm going to be your host for today's webinar. And our presenter today is our Head of Biology, Kersti Alm. She's going to tell you how to monitor and quantify cell morphology. During the webinar, please ask your questions anytime. We will answer them at the end of the webinar. So now I give the word to our Head of Biology, Kersti Alm.
Okay. Good morning. Thank you, Laura, for the introduction, and thank you everyone who's listening. I aim today to talk about why you should study cell morphology. Here to the left, you can see a movie showing MDA-MB231 cells. I haven't started it yet. And there you can see that even in a homogeneous cell culture, as this is supposed to be, cells have different shapes. And once they start moving, the shapes will differ every time you look at them. It's different. It depends on cell stage, cell density, cell movement. And when you study the morphology, you can tell a bit about the cell's life that you wouldn't know otherwise. I'm going to talk also about how we can measure cell morphology using the HoloMonitor, and how these morphology measurements can be used to study different aspects of everyday cell life, like cell differentiation or treatment with different substances. That's all part of a cell's everyday life. To start with, we can look at different natural occurrent processes such as cell division. I'm going to make a deep dive into this. But also like cell density, cell movement, everything affects the cell morphology. If you look at the pictures here, you can see a yellow framed cell, and you can see how that morphology of that cell changes over time. What you do with the HoloMonitor is that you also measure this. You get hard numbers on the cell morphology. So the first one is the area. You can see how the area of that cell changes. First, it's rather large, then the cell will contract. And after cell division, each cell has a very small area. And then the cell will flatten out again and the area will be large. Looking at the thickness of the cell, you can see that the thickness goes from rather thin, 3.4 micrometers, to almost twice that when the cell divides, and then you can see that the cell flattens out again. You can look at the cell volume and see how that shifts over time, how the volume is halved after the cell division. And you can also see this irregularity of the cell, how that shifts. A very round, smooth cell has an irregularity of almost 0. You can see the rounded up cell ready for division with a very close to 0 value. While a cell that's flat out and growing has a much larger value. Taken together, even without the pictures, the morphological parameters, they tell a story about the cell. Okay, so how do we then create these pictures? The instrument we use is a HoloMonitor M4. You can see that to the left in the pictures. That works by measuring how much the light is delayed when it passes through the cells. It's called a phase shift. That's what we measure. Then a computer will convert those measurements into pictures of the cells, like the one you see here. It's all label-free. It's all no phototoxicity. It's all very cell-friendly. But every picture I show you from here on is generated based on the phase shift of the cell, based on this little delay of the light. Every color you will see in every movie and every picture I show, we put on to show thickness of the cells. In the example image here, we got yellow as the thickest part of the cell and a purple as the thinnest part of the cell. If you want to learn more about the technology, you can watch the first PHI webinar, Label Your Tubes, Not Your Cells. So the good thing about using the phase, oh my God, what happened to that poor guy in the corner? The first thing you will notice about using the phase shift like we do is that you can get three-dimensional images. That is, we get the number on the thickness of the cell as well. In the left image here, we show the profile of the cells covered by the blue line. You can see there's a clear difference of the cell's thickness and the background levels. This makes it very much easier for the software to segment out what is cell and what is background. Based on that, we get all the morphological data. And you can get results from a single cell. Just measuring one single cell, you get a whole bunch of data. Next step, oh my God, there's one little guy again. They're all over the place.
Those are PHI monsters.
So you also get a lot of data from a single image with many different cells in. Then you can start comparing. Here, for example, the area of the cell is compared with the thickness of each individual cell. You see it in a scatter plot. Or you can get all the results from over time. You get, for a lot of time points, you get the results and you can see what happens over time. We call that the kinetic data. Altogether, we have all these morphological parameters presented by the HoloMonitor. I've talked only about the few, the area, irregularity, optical thickness, and the optical volume. But there are so many more. And all these parameters are described in the addendum to the handout. So look there if you're more interested in more in-depth descriptions. When we present these parameters, we do it through different assays that we have in our software. So just using the cell counter, you will still get cell area and volume. You just make a quick cell quality control, you get measurements of volume, area, and diameter of your cells. Or you do a dose response assay, and you get the cell numbers, obviously, but you also get the cell volumes. So there we utilize only a few of the parameters. For an in-depth usage of parameters, we have the cell morphology and the spatial cell tracking softwares. In the cell morphology, you get more than 30 cell parameters and can compare with different cells. And in the spatial cell tracking, we follow the cells over time, and we can see how all these parameters shift over time. I'll give you here an example of how to use the HoloMonitor. So step 1, capture images. 2, the computer will find out what is cell and what is background and will present the data in a graph. So to the left here, there is the control image showing untreated L929 cells. You see the color scale, so the really thick are yellow and the really thin are blue. To the right, you see what happens after 48 hours of colcemide treatment. See the cells round up and they become really large. But they're not as thick as the untreated control cells. You can see there are many kind of yellow cells up there, rounded, prepared to divide. And we can present all that data in graphs, scatter plots, or you get it as Excel sheets. In my example here, the yellow markers in the scatter plots are the control, the T0, and the blue ones are the T48. So the higher, up in the higher right corner show the control cells. They haven't really changed much in the morphology over 48 hours. But looking at the lower diagram, you can see that the colcemide treated cells, their morphology change. For each marker in the diagram, you click it and you get the corresponding cell. You get the data for each individual cell and you get all the population as well. We have protocols showing how to do this if you want to know more. So the other way to do this, instead of just capturing pictures at discrete time points, you can capture beautiful movies showing what actually happens to the cell, not just in the beginning and the end, but during the entire experiment. To the left, there are the control cells, and to the right, there are colcemide treated cells. And you see how they slowly shift and become really different from the control cells. They still round up and try to divide, but they are not really successful when it comes to cell division. And that's why they become larger and larger. So what we can do with this is to track each individual cell in that movie over time. So I picked out a nice cell here. It's treated with colcemide. As you can see in the row of images, time 0, time 8, time 18 hours, 26 hours, 38 hours, and so on, the cell morphology shifts a lot over time. If you had just the starting point and the end point, it wouldn't give as much information as having all the in-between steps. In the upper graph, you can see how the volume of the cell shifts from 0 to 48 hours. You can see that it kind of raises, the cell gets increasingly larger. But looking at the area in the lower graph, you can see that every now and then the area will be definitely much smaller. The cell will round up, and that corresponds to the pictures in the row below. You see the rounded up cell at time 8 hours. That's when the area is very small. So you get all these data over all time, both for the individual cells and for the population. If you want to know more about the cell tracking, please watch the third PHI webinar where you follow every cell step. So far, we have looked at the morphology and how the data can be created. Also talked a little bit about why, but how can you use it? How can you apply all this? I will show 4 examples of published papers that we have, but I think first Laura has something for you.
Yes, I think it's a perfect time to do a short break, and we would like to know if you know what is the shape of your cells after learning so much now that Kersti just talked about. Okay, so we got the results, and we actually see that most of you think your cells look like fried eggs. Are you guys hungry for breakfast or like thinking about lunch now? I don't know. Well, thank you very much. It was really interesting to see how you think your cells look like. And now I'm giving back the word to Kersti.
Okay. Very happy. I hope you managed to get a short break here now before I start talking about cell state classification. Dr. Kamlund and colleagues, including me, actually, as you can see in the author names, we have been quantifying EMT, that's epithelial to mesenchymal transition, and we used the, I can't really pronounce this, NMUMG cells, they're mouse mammary glands, because they are well known to be induced to transfer from epithelial to mesenchymal when you add TGF-beta to the cultures. So if you look at the upper left cell image, you can see a very epithelial structure, how the cells are growing. The morphology is clearly epithelial. If you look at frames C and E and F instead, the cells have been treated with TGF-beta, and you can see that they now have a more mesenchymal morphology. So Dr. Kamlund, she correlated 25 different morphological parameters with this transition. And in the end, she and Dr. Judson picked out 4 independent parameters that when they were weighed together, they would give you a classification. They would give you an EMT score, how the cells were changing. So you can see control cells have a large score, while mesenchymal type or epithelial type cells, while mesenchymal type cells had a much larger score. And then they applied that to different treatments, like you get the TGF-beta and you get some inhibitor, you get just the inhibitor. And they could see that the EMT score worked out well to classify. What they also noticed was that it's not really easy to just set a cutoff for everything. Every different kind of cell type will need its own cutoff. So you need to set up a control and a treated. You can't just go in and say this is valid for everything. But they show that with only 4 morphological parameters, no labelings, no stains, you can see and correlate to how the transition between epithelial to mesenchymal is progressing. The cell morphology can also be used to study cell differentiation. So here, Dr. Zhang and his colleagues have studied HL60 cells, which were derived from peripheral blood leukocytes. And they used retinoic acid to turn these HL60 cells from normal to N1 type neutrophils. And then adding TGF beta 1, they turned the cells further into type 2 N2 type neutrophils. And could see that using only the morphology, they could see that a lot have happened. If you look to the graph to the left, you can see that the HL60 cells, they have a volume about 1,000 cubic micrometers. And when they turn into the N1 type neutrophils, it's almost half the size. Or when they then consecutively go into the N2 type neutrophils, the size increases significantly. If you, in addition to that, also study how the cells move, you get a very clear definition of what is normal and N1 type and N2 type neutrophils. So you see the HL60 cells, they don't move much. The N1 type neutrophils don't move that much. But N2 type neutrophils, they move a lot. So even if the increase in size is not as large or as defined as the decrease for the N1 cells, if you combine it with the cell movement, you will know for sure in what stage your cells are when they differentiate. Dr. Makdasi and colleagues chose to look at ricin. This is a poison. It's really, really poisonous. It just takes like 2 grains, corresponding to 2 grains of sugar to kill a grown-up person. It comes from castor beans, and it inhibits protein synthesis. I kind of like that little detail. Dr. Makdasi tried 6 different concentrations, doses of ricin on her cells. They used HeLa cells. And by studying the response, the cell response over time, they could see that the effect of the higher doses of ricin were already after 1 to 3 hours, they could see the first responses. You could see an increase, a change in area. You could see a change in irregularity. You get starting to see a change in the length of the parameter already after 1 to 3 hours, just looking at the morphology. And through one single experiment, they choose to study 8 different parameters, telling how the cell progressed through the treatment and how the different doses affected the cells. And the last paper I want to introduce to you is from Dr. Barker and colleagues. They showed that they could differentiate between 3 types of regulated cell death by looking at only 4 different cell morphology parameters. That's kind of cool. No labeling, no staining, just these 4 parameters. So they started out by treating cells and made them go into ferroptosis, apoptosis, or necroptosis with 4 different treatments. And then they looked at the optical thickness of the resulting parameters here. They started with the optical thickness and could see that they could separate out really thick cells and really thin cells. Then they already had the necroptosis sorted out, because necroptosis cells are thicker than the others. Then they went to the optical volume, just straight off the volume of the cells. And they could see that when the volume was smaller, they had ferroptosis. And with the larger volume then, they combined that knowledge with a phase shift, standard deviation. And they could sort out not treated cells from the apoptotic cells. So in a 4-step classification. Just looking at morphology, they could say, this is that kind of cell death, this is that kind of cell death. I think that's kind of impressive. So, in short, cells change shape all the time. And these shape changes can be connected to example given cell differentiation, cell death, or different treatments. Data, both the single cells and cell populations, are needed for telling the full story. And once you have data in the HoloMonitor system, you have morphology, you can connect those data with motility or migration, or there are so many combinations you can do to enhance the use of the morphology. And all this is label-free. Okay, I think I talked at length about morphology, and I will hand now over to Laura again. Thank you.
Thank you very much, Kersti, for your very in-depth and very pedagogical presentation. So now we are coming to our Q&A part, and we got many questions. So thank you everyone who asked them. Since we're running a bit out of time, we're just going to answer a couple of them, and everyone else will get their answers via the email after the webinar. So first question is from Sarah from Edinburgh University. What does the morphological parameters mean? There are so many. Are they all relevant?
Okay, actually, that is a huge scope on that question. So for what the parameters mean, every single one of them, I will actually refer you to the handout, because there is a thorough description of every parameter. And there are so many, are they all relevant? That's a very, very interesting question. Thing is, a lot of them are obviously relevant, like changing area or volume or thickness or, irregularity. All those are obviously because you can see it directly. Other ones are more obscure, but are still very interesting. And because holographic microscopy is a rather new science still, we are not sure what each parameter corresponds to, biologically speaking. We are not sure when the hull convexity, for example, which is the shape, the dome shape of the cell, when that shifts, what biological occurrence is that corresponding to? We don't really know that yet, but we hope that you listeners will be able to tell us in a few years.
Thank you. Then we have another interesting question from Ulf from University of Oslo. He asks, can I measure morphology on all type of cells?
Well, I would say yes. You can use cell lines, you can use adherent cells, you can look at suspension cells, you can look at primary cells. There are physical limits. We have to be able to fit them into the microscope, but otherwise we can measure most kinds of cells, yes.
Okay, and since we're running out of time, we have time only for one question, and it comes from Germany. Lisa from Berlin is asking, can the images from the morphology be used for other assays, or does she need to redo the experiments?
Usually you can pull out much more data from each image. It depends on how often you capture the images. If you have images captured close enough, you can measure cell motility. But we need to be able to follow the cells rather closely to do that. Otherwise, you can always get parameters like cell numbers, cell count, confluence out from every picture. So yes, you can mine a lot of data from each picture.
Okay, thank you very much. Thank you, Kersti, and thank you everyone who asked the questions. If we haven't answered your question, we'll definitely do that via the email. If you have more questions after this webinar and you would like to contact us, please go to our website, write us an email, or connect with us and follow us on our social media. Thank you very much and have a nice day. Bye.
Bye.